I stand between you and lunch. So I think what I'm going to do, since Colin covered some of the topics really well this morning, if there's redundancy, I've left it on here so you can look at it later. There are three things that I'd like you to walk away from today if you walk away with nothing else from the combination of Ivona and my lecture. One is that life in the ocean depends on phytoplankton. No phytoplankton, no life. And that's because they're photosynthetic. The second thing is that when we say phytoplankton, we're really lumping things that are totally dissimilar in terms of their genetics. They're taxonomically and functionally diverse. I'm going to break this group into two functional groups, students and instructors. Do you feel comfortable being lumped together? Have I captured your diversity by saying you're students and that's all I need to know about you? I don't think so. They found out, there's an article that was published in Nature last month that said diatoms have this whole other enzymatic pathway that when they're under stress, they change their metabolic rate. And so those of you who are physiologists know about the von Bernoulli equation in terms of respiration is scaled to size. They step out of that. They have a different genomics that allow them to change their physiology. So some of the things in terms of the challenges of functional groups is, are you comfortable being students? And are you comfortable by being faculty? And I've categorized you one way. Is that a good enough function? So it's a challenge. So life depends on them. They're really diverse. And then what I'd like to focus on is that phytoplankton are the only organism on this in the ocean that you can measure in a teaspoon, a drop of water, an ml, and you can measure it on the scale of the globe. So if you want to do zooplankton ecology or fish ecology, you're going to model it based on something related to phytoplankton because you can't measure fish from space. Maybe you can go do some acoustic survey someplace, but I think that it's a really long way and maybe never that you're going to measure acoustic reflection of fish from space. So phytoplankton, we can measure from the micro scale to the global scale, and that's due to chlorophyll. And so I'm going to touch on that and also follow up a little bit on some of the things that Ivona said in terms of how you measure phytoplankton. You saw this slide yesterday in terms of going from the satellite. This is a NASA-sponsored course. The interaction with radiometry, the in-water optics, and that gets us to the surrogates or the proxies, and then to the real entity. And very often, how you define the real entity, going back to, do you want particulate organic carbon if you're going to be measuring carbon in the deep sea? Maybe that's what you want as POC. Maybe if you want harmful algal blooms, what you really want is to be able to measure cells. So there's this sort of trajectory going from the satellite to the real entity, and then scaling back up to the ecosystem to some of the real fundamental problems that we're facing today, like the Pacific Northwest with that pool of warm water, with the Pseudonychia blooms, with the Coccolithophore blooms, with everything else. So this is sort of the world that we live in. And putting that into a different way, in terms of how we use optics to study biogeochemistry, we go from a variable that interacts with light. And then we measure a volt or a digital count. We turn that into an IOP or an AOP. And then for those of us who are more ecological, we turn that into a proxy. And what we're trying to do is get to whatever the variable is, the real thing. And so there's sometimes a long way between what we're actually measuring and what we're trying to get at. And I think that is in spades for this functional type. So anyway, that's just another way of thinking about things. So how are we going to measure phytoplankton? We can go out and we can count them, and we can do it with simple tools, historical tools, new tools. We can gene sequence them, still not really quantitative. Or we can use optics, because optics are related to absorption. So that's, for phytoplankton, somewhat unique. It's related to scattering, which is phytoplankton and scattering. It's not unique. We have other things that scatter. And fluorescence, in terms of specific optical signatures, fluorescence is unique. So phytoplankton interact with light. That's the basis for these optical properties, like phytoplankton mass, particle number, etc. So particles scatter light. Phytoplankton are particles. Pigments absorb light. Phytoplankton are photosynthetic, therefore they have pigments that absorb light. And several pigments, chlorophyll A specifically, and phycoerythrin, also re-emit light as fluorescence. And what I want to do is go through several things. One of them is, how have we measured chlorophyll? And someone made this point yesterday, I don't know if it was Colin or Emmanuel, that sometimes we measure multiple things. We're trying to get at one thing, for example, phytoplankton, but we're really measuring parts of it. It's sort of like feeling the foot of the elephant and try to project the elephant, or feeling the tail of the elephant or the trunk of the elephant. And so there are various measures. And sometimes by using multiple ones, we can actually get closer to what that entity is. And then I'm going to end with physiology can sometimes make our life difficult because it changes some of the relationships between the optics and the real entity. Plasticity, so physiological plasticity, morphological plasticity, is essential for survival, and it's a real annoyance for us. But you have to know about that in order to make use of that and not be defeated. So Colin talked quite a bit about chlorophyll and about pigments. So there are three basic types of pigments in phytoplankton. The chlorophylls, everybody's got chlorophyll A, or Prochlorococcus has a variant of it with a slight change in the chemical structure that gives a slight shift in its absorption spectra at the Sorey peak. So there's chlorophyll A, everybody has chlorophyll B or C. Some have B, the greens, many have C. I don't know of anything that has both B and C. The carotenoids, which can be divided into two types, the light harvesting, which are accessory pigments to chlorophyll, and the light protecting, or the photoprotective carotenoids. And many of them overlap with the same spectra, but both of them absorb light mostly in the blue-green and the green. And then the third type that are found in cryptomonads and cyanobacteria are the phycobilins. So if you do HPLC analysis, you're not going to get the phycobilins unless you're doing something that's specifically water-soluble, because they're water-soluble pigments. They're not extracted in a polar solvent. So those are the types of pigments. I'm going to go through this fast because you've got it in the PowerPoint on the web. But here again is the pure chlorophyll A, the structure of chlorophyll A with a phytol tail, a porphyrin ring, magnesium at the center, 4 nitrogens in the porphyrin ring. Colin talked a lot about changing the structure by adding acid and losing either magnesium or the phytol tail, and there's a shift in the peak, and there's a reduction in the red peak. So there are some changes with degraded pigments. This just backs up Colin's comment about the environment makes a difference in terms of how the pigment interacts with light. And these are 6 different solvents, and you can see that there's a change in the absorption efficiency, as well as some changes in the wavelength peaks. And again, I'm going to go through this fast just because. Chlorophyll A brackets the absorption spectra. So the Sorey peak is the furthest to the blue, the high energy absorption, and it's the furthest to the red. In between are chlorophyll B, so it's shifted to the red. Chlorophyll C is shifted to the red. And so that allows a transfer from absorption of higher energy to ultimately the red, the Q band absorption for chlorophyll A. So there's a reason for the absorption spectra evolutionarily in terms of any light that absorbed will be ultimately transferred to the Q band absorption for chlorophyll A It's the lowest energy, and so energy is transferred downhill. Carotenoids come in both photoprotective and photosynthetic absorptive. They're characterized by a lot of bonds, and you can think of them for the photoprotective is that they're sacrificial. So if any of you have done enzyme analysis, you add bovine serum albumin so that any bad enzymes chew that up instead of chewing up whatever the enzyme is if you're doing an enzymology test. I did my postdoc doing enzymology, but it's sacrificial to free radicals. And so the basic characteristic of carotenoids are these very long bonds. And they tend to absorb in the blue-green and into the green. And a lot of them have overlapping absorption spectra. And here are the different flavors of the phycoerythrin pigments that you see in the ocean. Phycoerythrin, it fluoresces orange. Phycoerythrin, which is, so phycoerythrin absorbs around 530-ish. It's got a broad absorption. Phycoerythrin is found in a lot of cyanobacteria in the deep blue sea, in the blue ocean, because it absorbs at 490. And phycocyanin you tend not to see so much in ocean pigments, except as an accessory in terms of ultimately transferring the energy to chlorophyll A. Here's a composite, I think from the web book. Did I steal that from the web book? With the absorption spectra of all of these different pigments. Not every species has everything, but the end result is something that looks like this. And so if you turn around and look out at the green grass, that has chlorophyll A and chlorophyll B. So it's a little bit further because chlorophyll B is over here, and chlorophyll B in the Q band is over here. But if you compare the two, what's the most predominant characteristic that you would say? If you're going to say in 4 sentences, what's the difference between this and this? Is this a test if you're awake? What's that? Right, right. So there's absorption. It's a hole right here. So there's no absorption. So if you put, dig up a plug of grass, put it in a jar, put it down at 10 meters in the Damariscotta River, would it photosynthesize? So here's the light spectrum in the Damariscotta River. Here's the light spectrum out in the Sargasso Sea. And in the Damariscotta River, because of CDOM, blue light is absorbed really, really fast. Red light is always absorbed because it has a high absorption coefficient. Water has a high absorption coefficient in the red. And if you go down, and the divers here, manual dives, you go down to 20 meters, the only light you're going to have is going to be green light, kind of greenish yellow light, because it's so CDOM rich. And so if you put an alga that did not have any accessory pigments in here into the Damariscotta River, it's not going to survive because there's a mismatch between the available light and the potential to absorb pigments. So green algae, people think absorbed, evolved when there were already halobacter, and halobacter have pigments that absorb in the green, and they use that to drive hydrogen flux so they can generate ATP. And so the evolutionary idea is that that spectrum, because that's the highest spectrum in terms of the light coming in, the solar energy, is that this was already taken. And so the green algae that evolved took up the leftovers. And so green algae can work in a terrestrial sphere, but if you put them into the ocean, where the local ocean here, where there's high CDOM absorption, there's not going to be light, so they're not going to absorb. So these accessory pigments are really critical for allowing phytoplankton to absorb wavelengths that are not available to green plants. So that was a race through pigments, and I'm a little bit off center here. What I'd like to do is just go through some ideas about how chlorophyll allows us to see phytoplankton all the way from the droplet to the entire globe. And so when we think about chlorophyll, and I don't know if Ivana had that on there, so if you think about chlorophyll, Ray Smith, some of you know the name Ray Smith from the literature, from the optical literature, and Ray would say, well, I measure chlorophyll, you know. Got it, this tube. It's chlorophyll, I'm measuring chlorophyll. So it's like there's this idea that chlorophyll is kind of freely floating in the water. And so how many of you know about the organization, you know, if I asked you to get up here and talk about the organization of chlorophyll in the cell, who would feel comfortable in volunteering to run up here and draw a diagram? So what's your idea about chlorophyll? So we're seeing something related to chlorophyll, and I'm going to put chlorophyll in quotes because there's chlorophyll B and C, and there are carotenoids. And so these images, we call them chlorophyll, but just keep in mind there are other pigments in phytoplankton that are contributing to absorption at wavelengths. So here's chlorophyll. This is 436, and this is 676. And so really, a lot of what we're seeing with ocean color is stuff that's absorbing in here. So I'm going to put chlorophyll in quotes. So here's the globe, and we're seeing something. These have been turned into chlorophyll concentrations. And so you've seen this already. Here's chlorophyll. Here's the phytol tail. Here's the porphyrin ring. It has a molecular weight of something around 946-ish. I may be off 20. But that's chlorophyll, and that's what in the lab on Friday you're going to extract, and you're going to put it into acetone, and you're going to measure it. But that's not what it's doing in the cell. This was very exciting about 10 years ago, where the structure of a protein that crosses the thylakoid membrane was first determined. And so there's this protein that goes across the membrane, so it's sort of like this. And you can think of it as a Christmas tree. And on that Christmas tree, there are various pigments that are associated, so it binds to the various pigments. So you don't have chlorophyll just sort of jumping up and down, you know, in the membrane. It is bound to a protein, and that protein, if the pH changes, is going to have a conformational change. And so if I'm spread out like this and I'm absorbing photons, am I going to absorb more photons if I'm like this?
Or if I'm like this?
So there are changes that happen in terms of the conformation of this transmembrane, transthylakoid membrane spanning chlorophyll binding protein. So chlorophyll is stuck to that, and then this forms what they call a monomer. And so you have this transmembrane spanning protein, which you can see if you squint. And so this is one complex with the protein and the pigments. And so it's got the chlorophylls and the carotenoids. And then this is a second one. And up here, this is a third one. And so these together form what they call a trimeric complex of the protein and the pigment. So this may not be what you think of when you think about chlorophyll. You've got the molecule stuck to a protein forming a complex, and then you have multiple of these trimeric complexes organized so that the energy that gets absorbed gets fed into a reaction center complex, and that's where the actual work of photosynthesis gets done. That's where a high energy electron gets ripped off of one chlorophyll molecule and transferred to another. So photosynthesis is actually a redox reaction, and it's enabled to happen because of this organized complex that's shunting energy in to the workhorse of photosynthesis, these reaction centers. So again, when you think about chlorophyll, you can think about these things also being able to move in response to a change in pH, which is mediated by changes in light. And then those complexes are, you can think of them as kind of globs. And this is a membrane. So you've got what they call a bilipid membrane. So you've got the lipid part and the protein part. And so these things can kind of move around. So I don't know a good analogy for, something squishy, floating and, moving, but it actually can move as there are changes that are related to the pH. So you've got these complexes here in the membrane, and the membrane makes up part of a chloroplast. So this is a chloroplast with lots of these membranes, and these membranes have these big units of chlorophyll that are bound to the protein. And so all of this is related to its functionality. And so here's a diatom ditylam brightwellii, which is pretty big, it's 60 microns, and these are chloroplasts in the, so the chloroplast has all these membranes. So now you have multiple chloroplasts. Here's a casinodiscus, all these little round globby things are chloroplasts, and they're organized in the cell. Here is the Lacyanema. These are individual cells, and all the bright red stuff are chloroplasts that have been illuminated with blue light and they're shining red light. So if we go from the individual chlorophyll molecule, we can scale up to the chloroplast, and then those pigments, whoops, I'm just going to go through this. Those pigments give us an absorption spectra, which then if the light is absorbed, Colin talked about the reflectance spectra. The amount of phytoplankton is going to change the reflectance spectra. The more phytoplankton, the less blue light. The less phytoplankton, the more blue light gets reflected to your eye. And that allows us to scale up to the globe. So going from that individual molecule all the way to the globe has to do with the absorption. But then, I think in the last week I'll talk about photosynthesis. I want to try to bring this back down in terms of the absorption related to the ocean color and how we come up with, again, it's like life of a photon that Kurt was talking about, but with a different scale in terms of the relationship. Okay, so that's trying to link a molecule to a global picture of phytoplankton. And it's really pretty exciting that you can actually see biology from space, and everybody else has to model based on phytoplankton. What I'd like to do next is just give a little bit of a history in terms of how people have measured chlorophyll. So has anyone ever heard of the Munsell color chart? If you're a geologist, you still use a Munsell color chart when you go out and tromp around in the mud. But actually up until the 30s, there was a color chart that people would take out, they'd filter a sample. And so tomorrow, you're going to do the filter pad measurement that Colin was talking about. And your eyeball will be replaced by a spectrophotometer. So it was a qualitative way. Have you ever shut your eye and opened one and noticed that the color is actually different? So how quantitative do you think the Munsell color chart was, with your very calibrated? But this was a semi-quantitative way to measure phytoplankton concentration by comparing it. And you had chromicity, as well as concentration. So that gave way in the 50s, where people went out and they took the filter, they extracted it in a solvent, and then they put it in a spectrophotometer. And they were able to use a series of equations. They're trying to get at chlorophyll. And so there's this fixation on, I can't spell when I'm standing at the board, chlorophyll. Is that what you really want? And so that's what they really wanted. Here this was related to a standardized color chart. Then they said, okay, we can extract the pigment in a solvent, and we can use a series of equations to try to remove the effect of chlorophyll C, the carotenoids, and come up with a concentration of chlorophyll. Let me tell you that a factor of 10 reduction in sensitivity, if you've done any chemistry, if you make a measurement with fluorescence, you can have 10 times less concentration than if you do it in a spectrophotometer. So you would stand at a filter rack and you would filter and you would filter and you would filter, and the filter would clog, and you would filter and you would filter. And so if you go into the NOAA databases, you can still get this data. And if you're trying to do a historical, has the ocean changed? Just let me tell you that when you're standing there filtering for a couple of hours, because there's a lot of stuff in the water, do you think anything happens on that filter? Do you think there's any oxidation on that filter? And then you're bored out of your gourd, and you finally get one of them done. You fold the filter, you put it in aluminum foil, you put it in a desiccator, you draw the vacuum, you put it in the freezer. The next one is done. So you take it out of the freezer, you fold the filter, you put it in the desiccator, and then the lunch bell rings. And you run down, you have lunch, and 2/3 of the way through lunch you go, oh, I left the chlorophyll desiccator out on the table, and it's now warming up. So if your advisor says, I think you should do a historical analysis of the chlorophyll in the Indian Ocean, just keep in mind that this is never going to be in a database, but I can tell you from talking to the people who did it, there's lots of problems. So the next advance was in the early 60s, taking that filter and throwing it in acetone and then measuring the fluorescence. And so that was much, much faster. And in fact, if you look today, here's the Southern Ocean iron enrichment experiment. You will see this method is really commonly used by most oceanographers. Now, it may not be good enough for NASA, but I'm going to tell you that 98% of the papers that measure chlorophyll will measure it this way. And we're going to do some comparisons with a couple of different techniques with that methodology. And then back again a little bit later in the 60s, a guy that I had the pleasure of knowing, Carl Lorenzen, took and started taking advantage of the fact that if you shine blue light on live phytoplankton cells, they re-emit red light. And so what he did is he took a garden hose and he put a pump on it, and you taped a long extension cord to the pump. You're on a ship, this is salt water, and they're lowering a pump, and you're pumping the water up and you're pumping it through a fluorometer. It was an electrifying experience. But the first continuous profiles of phytoplankton were done from the side of a ship with a garden hose and a pump, pumping things through a fluorometer. Now we've gotten much more sophisticated, and in Friday's lab you'll be doing a lot of work with different types of fluorometers, and now it's really common. Here's some old data from 10 years ago collected on a glider in Monterey Bay, and you can see the features in terms of tidal focusing and defocusing, forming a thin deep layer, and then relaxation in terms of the tidal pumping. So this has really allowed us, as Ivana was saying, to measure some aspect of biology on the same space and time scales as the physics. And this has really kind of changed the way that we think of things. Colin talked a lot about this morning in terms of the quantitative filter technique. This is from the subpolar North Atlantic, a total phytoplankton absorption. We extract the pigments with methanol, and you'll do that in the lab tomorrow. And the spectra that remains is the non-algal pigments, and their composition can be many. Take the different spectra, and you come up with the phytoplankton absorption, to which a beta has to be applied. And so there is still some uncertainty in all of that. And Colin talked again this morning about the composition of absorbing material. Phytoplankton is one of them. And here's that green, kind of nice light green. And the magnitude of the different absorbers give rise to the various reflectance spectra that let us then go to something that we dearly, dearly, dearly want to call chlorophyll. You know what chlorophyll is now. Here's another methodology Ivana talked about. And I'm not going to belabor this, but HPLP pigments, and with the in-water spectrometer, you'll do Gelb stuff today, dissolved organic material, yellow substance. Tomorrow you'll do particles. Here's chlorophyll in the absorption spectra. This is with an AC9 from some of Colin's data. And again, here's another indication of chlorophyll. So we've gone from the Munsell color chart with the calibrated eyeball up to in situ spectrometers and fluorometers that can give us a much more defined spatial and temporal view of biology in the ocean. And it's all based back one way or another to chlorophyll and to its accessory pigments. And I have one more set of slides, and I'm just wondering, I feel like I could race through this, but I think that it's actually worth taking maybe 10 minutes, and I don't think the kitchen will be very happy with us if we get up there late. And I want to talk about Mobley's law of conservation of misery, and I think it's worth maybe. Yeah, do it. Yeah. So do we want to do 1:30? Is that okay, or?
Or we can come back to 1:20 if we feel that.
Maybe 1:15. Would that be okay? I think Kurt actually enjoys this lecture because he's got his own stories on conservation of misery.
Just a quick announcement. I found out recently that a couple people can't get to the FTP site. I'm really sorry.